So I want to talk about a disease that mosquitoes carry called malaria and how we might actually solve the malaria problem in the world. Malaria is actually a parasite that lives inside the mosquito, and if you get malaria, the parasite gets into you and it burrows into your red blood cells, and it can cause them to lyse, so it'll kill the red blood cells. Now with biotechnology, we can take a mosquito, do some bioengineering, and we can change a gene so that the mosquito is resistant to malaria. So this in our little story is going to be a genetically engineered mosquito. Now we're going to take this one mosquito and we're going to let it loose into the wilds. We are going to let it breed with a normal mosquito. So here's our genetically engineered mosquito. I'm going to have it mate with a female. So the female does not have the gene. So there are her gametes, right? So we got a Punnett square here. So this male, he's going to drop down his alleles, which all have the fixed gene. So we're going to put his alleles like this. Allele for the female, right? And as you can see from our previous work with Punnett squares, we now have F1s, right? We start with the family, we have the parents, we do the Punnett square, we got one baby that's a carrier, another baby that's a carrier, a third baby that's a carrier, and a fourth baby that's a carrier. I'm going to take one of these F1s, let's say, how about this one, and we're going to create the F2 generation over here. So here's going to be the male, and these popsicle sticks are going to represent a female out in the wild, like this. And just like before, the male's going to drop down his alleles like that. So that's the chromosome with the genetically modification. So he drops down his alleles like that. Then the female is going to push over her eggs like that. So we're making their F2s. Now you can see in this family, half of the F2s are carriers and the other half are normal. So over here we have our initial parents. The little m is the male that had the genetic modification, and the big M is the wild female. So in this first cross, as you know, we get 4 F1s that are all carriers. Now let's pretend we let this mosquito loose in a population that had, say, 1 million mosquitoes. Now, right now we have 4 out of 1 million that are carriers. We go over to this next little picture, and this one F1 here has a little family, but I have 4 F1s. So I'm going to have 4 Punnett squares in this stage. And each one mates with a mosquito in the wild that's normal. So when we go through the Punnett squares, and this is the second one I showed you, we have 2 out of each Punnett square that are carriers. So 2, 4, 6, 8. So now in the F2s, we have 8 mosquitoes out there in the wild that are carriers. But I'm going to take these 8 mosquitoes that are carriers and bring them over and make the F3s. So again, this is out in the wild. So these 8 Punnett squares are going to represent the new families, and it'll be just like before, where each Punnett square is going to have half the offspring be carriers. So when you add these up, we get 16 out of the F3s are carriers. Then we're going to go and do the F4s. So I just take these guys and I make my F4s, and it turns out that we have 32 that are carriers. And then by the time I get the F5 generation, we have 64 mosquitoes out in the wild that are carriers. Now we haven't solved anything yet. These mosquitoes can still give you malaria. But what if we could use a gene drive? Now we are going to introduce a mosquito that has a gene drive. Now a gene drive is a DNA code. It's basically CRISPR installed in the genome. So once you get the gene in there and then you install the gene drive, watch what it can do. So this male mosquito is going to be released into the wild like that. It's going to mate with a normal female. But the gene drive takes over, so we drop his chromosomes down like this. All right? And he has 4, so we put those there like that. Now the female, she passes over normal chromosomes like this. So we're making our F1s. Everything's looking like it did in the first time, more or less. Once fertilization happens, the CRISPR part, which is the gene drive, copies this gene, that's the GMO modified gene. It walks or drives over to the other chromosome. It finds the exact spot. It snips, cuts, edits, and it converts the female's chromosome, changes the gene, and adds the gene drive.
Ooh, that's sneaky. That is sneaky.
So that happens here, it happens there, it happens
Slick
there.
as goose grease.
Hey, Sophie.
It is slicker than goose grease. And this technology is not happening in the lab. This is happening inside the cell in the wild. These cannot give you malaria because they have the gene and they have the gene drive. So this is really different. Now we're going to go over to the F2s. So I'm going to take one of these 4. They're all the same. So I'll use this one. So there's the male, and the female is normal. So we put her out here like this. And there's the other normal female. Now the male's going to drop down his gene drive and fixed chromosome. There's 1, there's 2, there's 3, and there's 4. The female sends over her chromosome. The gene drive takes over, it converts it into just like his. It adds the new gene. So this will be just like before, that all 4 of these not only have the gene for malaria resistance, but they have the gene drive. Remember, this is what we had the first time.
Flip it.
Oh!
Okay, let's start out with the gene drive. This parent not only has the gene, but he has the gene drive installed in each of his chromosomes. He mates with a normal female. All of the F1s, 4 of them are malaria resistant. One of them flies over here to make this family, another one to make this and this and this. We go through the F2s. Now we have 16 that are resistant to malaria. They make over the F3s. We have 64 that are malaria resistant. By the time we get to the F4s, we have 256 that are resistant, and 3 months later, we have 1,024 mosquitoes flying around the swamps that are all resistant out of 1 million.